The problem with the majority of cells in our body is that they are not in direct contact with either the digestive system, which is in charge of supplying nutrients to the cell, is not in contact with the outside world, which is in charge of getting oxygen into the cells, or is capable of taking carbon dioxide away from our body. This is where the cardiovascular system comes into play. The cardiovascular system provides a network or a highway system that is in contact with every cell within our body. This allows us to transport nutrients to the cells and get rid of waste from the cells. When you think of the cardiovascular system, you need to remember that there are three components to it. The first is the heart, which is a pump. The second is the blood vessels, which you can think of as a long hose, or a highway system, which connects every cell to the necessary organ systems responsible for maintaining homeostasis. The third component is the blood, which you can think of as the cars or delivery trucks. The heart is composed of 4 different chambers: the right atrium, the right ventricle, the left atrium, and the left ventricle. The atrium can be thought of as receiving chambers. They receive blood from the systemic circulation and the pulmonary circulation systems. In contrast, the ventricles are considered the pressure chambers. They generate a force to deliver the blood to both systems. The cardiovascular system has 2 different components. The systemic system is in charge of delivering oxygen and nutrients to the cells and picking up waste material. The pulmonary system provides the body a way to pick up more oxygen and drop off unwanted waste like carbon dioxide. Each side of the heart is responsible for receiving blood from one system and ejecting blood to the other system to continuously provide a cycle of delivery and refreshing. Specifically, a drop of blood would return to the right atrium after coming back from the systemic system. It is low in oxygen and high in carbon dioxide. That blood would then travel to the right ventricle, where it would be pumped to the pulmonary circuit. Once in the lungs, the blood would drop off carbon dioxide and pick up more oxygen. The blood then returns to the heart, specifically to the left atrium, is passed to the left ventricle, and then pushed forward back to the systemic tissue to deliver oxygen and pick up more carbon dioxide. In order to move blood throughout the body, the heart has to generate pressure. This pressure results in a pumping force to move the blood from one place to the other. Imagine having a bucket of water and placing a hose in the bucket. That water will remain in the bucket because there is no force to move the water into the hose to the other side. The heart receives blood and is able to generate pressure, which can then be used to force water into the hose. In order to generate pressure, we have to be able to fully close each chamber. This is accomplished through valves that are placed in between the chambers. In between the right atrium and the right ventricle is the right atrioventricular valve. In between the left atrium and the left ventricle is the left atrioventricular valve, AV valve for short. In between the right ventricle and the pulmonary system is the pulmonary valve. In between the left ventricle and the systemic system is the aortic valve. There is no valve from the systemic system leading to the right atrium, and there is no valve between the pulmonary system and the left atrium. The atriums are consistently and constantly receiving blood from both systems, which creates pressure in the atrium. Valves function like pushing a door open. When you generate more force than what's on the other side of the door will swing open. If you do not generate more force than what is on the other side of the door remains closed. The same is true for valves. The heart contracts in unison. Both atriums contract simultaneously and both ventricles contract simultaneously. Therefore, both AV valves open simultaneously and close simultaneously, and both semilunar valves, the aortic and the pulmonary, open simultaneously and close simultaneously. This ensures efficient pumping of blood throughout the heart. When atrial pressure becomes greater than ventricular pressure, the AV valves will open. The AV valves will remain open until ventricular pressure becomes greater than atrium pressure. Once ventricular pressure becomes greater than atrium pressure, the AV valves are forced shut. There is a brief period of time when both the AV valves and the semilunar valves remain closed. This is because ventricular pressure has not overcome systemic pressure. When ventricular pressure becomes greater than systemic pressure, the semilunar valves are forced open. They remain open until systemic pressure becomes greater than ventricular pressure, at which time the valves close again. This alternating generation of pressure is what forces the valves open and close and allows for our heart chambers to fill and eject blood efficiently. The valves are prevented from allowing backflow because they are securely attached in the ventricles by chordae tendineae, which are attached to papillary muscles. These hold the valves securely in place, which prevents them from opening up back into the atrium. Neurons have a negative resting potential because of a high concentration of potassium inside the cell, along with negative anions. There is a large concentration of sodium and calcium outside the cells. This creates an electrochemical gradient that the cell can use to create action potentials. When the neuron receives enough outside stimulation, it reaches threshold. Threshold causes a rapid change in membrane potential from negative to positive. This is accomplished by sodium ions rushing into the neuron. The cell quickly returns to resting potential by the opening of potassium channels, which allows potassium to rush quickly out of the cell, bringing the charge back to a negative. The extracellular fluid within the cardiac cells is the same as neurons. This means that there is a high concentration of potassium inside the cells and a high concentration of sodium and calcium outside of the cells. All cells maintain a negative resting cell potential. In the heart, there are two types of cells: autorhythmic cells and contractile cells. The majority of cells are contractile cells. Autorhythmic cells differ from both contractile cells and neurons because they do not need outside stimulation to reach threshold. They slowly drift from a negative charge to a less negative charge until threshold is reached. Once threshold is reached, an action potential is generated. This drift from a negative resting membrane potential to threshold is accomplished by funny channels. Funny channels are essentially sodium voltage gated channels. As the autorhythmic cell becomes negative, these sodium channels open, allowing sodium to rush into the cell along its electrochemical gradient. These channels remain open until threshold is reached. When threshold is reached, calcium channels open, allowing calcium to rush into the cell along its electrochemical gradient. When peak positive potential has been reached, potassium channels open and potassium rushes out of the cell, reestablishing the negative membrane potential. Again, when the negative membrane potential has been reached, this triggers the funny channels to open again, which results in the regeneration of an action potential. All of this is done within the cell and requires no outside stimulation from neurons or hormones. Autorhythmic cells and contractile cells are connected to each other via gap junctions. These gap junctions allow an action potential in one cell to spread to the other cells it is connected to via gap junctions. Gap junctions allow the heart to efficiently contract as well as rapidly contract. It decreases the amount of stimulation the cells need in order to generate their own action potential and thereby their own contraction. The contraction of the heart begins in the sinoatrial node. This is located in the right atrium. From the sinoatrial node, the impulse from the action potential quickly spreads to both the internodal pathway and the intraatrial pathway. The intraatrial pathway allows the generation of action potentials in the left atrium. The internodal pathway allows the impulse of action potentials to spread towards the ventricles. These action potentials result in the contraction of cardiac contractile tissue to generate a contraction of muscle to move the blood from the atrium into the ventricles. Once the action potential spreads to the internodal pathway, it will be received by the atrioventricular node. There is a slight delay here to allow atrial contraction to be completed, which allows fuller filling of the ventricles. From the AV node, the impulse spreads to the bundle of His, which breaks into a right and left bundle branch. These bundle branches allow action potentials to rapidly and evenly spread between the left and right ventricles. The right bundle branch and left bundle branch connect to the Purkinje fibers, which wrap around the apex of the heart back up towards the atrium. This allows for contraction to force the blood upwards to the semilunar valves. Each autorhythmic cell step has its own rate of action potential generation. The sinoatrial node depolarizes at the most rapid rate of 70 to 80 beats per minute. The atrioventricular node depolarizes at a rate of 40 to 60 action potentials per minute. The right and left bundle branches and Purkinje fibers depolarize at a rate of 20 to 40 beats per minute. The benefit of having different rates of depolarization are that the heart would continue to contract even if one of the autorhythmic cells failed. For instance, if the sinoatrial node failed, the atrioventricular node would take over contraction of the ventricles, which would still allow for the heart to pass blood into both the systemic and pulmonary system. The autorhythmic cell with the fastest rate of depolarization controls the heart rate of the rest of the tissues. If the sinoatrial node depolarizes, that depolarization will spread to all other autorhythmic cells, interrupting their normal rate of depolarization. Therefore, if the sinoatrial node is functional, all the other autorhythmic cells will take on the rate of the sinoatrial node of 70 to 80 beats per minute. If the sinoatrial node were to fail, the atrioventricular node would take over the rate-setting pace of the heart. The right and left bundle branches, as well as the Purkinje fibers, would take on the rate of the atrioventricular node of 40 to 60 beats per minute. The neuron starts with a resting membrane potential of approximately negative 70 millivolts. When it has received enough excitatory synaptic potentials from other neurons, it reaches threshold of negative 50 millivolts. This is done by the opening of sodium channels to allow sodium to rush into the cell. When threshold is reached, more sodium channels open, allowing a rapid change of charge. When 30 millivolts have been reached in the neuron, potassium channels open, allowing potassium to rush out of the cell, bringing it back down to a negative membrane potential. Contractile cardiac cells follow a similar pattern as a neuron. It has a negative resting potential of negative 90 millivolts. When it receives a stimulation from gap junctions or other stimulating input, it will reach threshold of negative 70 and then rapidly change to a positive millivolts. This rapid change is accomplished by the opening of sodium channels, which allow sodium to rapidly rush into the cell. The main difference between cardiac contractile tissue and neurons is step 3 in the diagram. This plateau phase is accomplished by the opening of calcium channels. The calcium channels open and allow calcium to come into the cell, which maintains this positive plateau of the action potential, prolonging its duration. After the calcium channels open, potassium channels then open and allow potassium to rush out of the cell, reestablishing its negative membrane potential.